great always to see old friends, make new friends, and learn something at the same time. It's all good. So I was asked specifically to, let's see, all right, talk on like the clinical utility of genetics and SJIA MAS. And the short answer is there's not a whole lot. So I'm going to talk about, because you'll hear other talks following me, and I do have disclosures. The HLH genes, or the hemophagocytic lymphohistiocytosis genes, and these are the genes that give you the familial form of MAS or HLH. That's a genetic cause where there's defects in the ability of natural killer cells and cytotoxic CD8 T cells to kill their target cells. And if we can go back a couple decades to Alexei and colleagues, Lisa Filipovich and colleagues in Cincinnati, they realized early on that in children with systemic JIA who developed macrophage activation syndrome, that their natural killer cells function was not ideal. And from that, they also identified one of these genes, MUNC13.4. It's confusing. It's also called UNC13D, so it's the same gene. Kids with systemic JIA and MAS in their cohort, 57% of them had this linked group of polymorphisms on that gene of the kids who developed MAS compared to only 8% of the children with systemic GAA, because we'd at least have a couple cohorts of kids with what we call systemic GAA, some kids who tend to get MAS over and over again, and some kids who really never get it or just get it once. Anyway, the kids who didn't get it had a much lower percentage, as did the healthy controls. And then in collaboration with some of the folks in Boston at the time, Bryce Binstead and colleagues, they had a patient who had actually a mutation in this gene. So these are the same genes that lead to the familial form, but this is single copy. So this is not biallelic. It's one chromosome with a bad gene. And if you send commercial studies or you do your whole exome sequencing, you're going to probably miss some mutations because mutations can occur outside of the parts that code for protein. And here's one that Alexei and Hermina and Grant had a child in Cincinnati who was one of these kids with systemic JIA, just kept getting MAS over and over and over again. They obviously knew what genes to look for, but they could never find a mutation until they looked in the first intron there. And then we collaborated with them and showed that it was a functional mutation that decreased expression by disrupting a transcription factor site. And then Voss and Fabrizio and colleagues in Europe reported on one of these genes, and only one of these mutations. They had multiple mutations, but this one specific mutation in perforin that changes an amino acid to a valine, which is common. In the US, it's in Caucasians around 8%, roughly similar in Western Europe, and they showed that in their cohort of children with systemic JIA who developed MAS, 20% of them had this one gene mutation, so not all the genes that are involved, but just one of these genes, and actually only one of the mutations of that gene, compared to 10% of the kids with systemic JIA who didn't have MAS. So this is all kind of saying, even if you have one bad copy, it suggests that maybe these genes contribute to the risk of developing MAS. And so I moved from Philadelphia to Birmingham 17 years ago, and we started getting consulted in the hospital on kids with systemic JIA and MAS, but other kids with MAS from other causes. And again, you can see the kids in pink or whatever that color is. The UNC13D gene and this perforin A91V gene would show up in these kids who get MAS. But you can see all these other HLH genes, RAB27A, syntaxin binding protein 2, they're starting to show up in some of these other kids who got MAS. And that's actually what Alexei again and colleagues, Ken Kaufman et al., and Lisa Filipovich in Cincinnati showed in a small cohort of kids, that around 30 to 40%, and this number shows up a lot in different cohorts, not just systemic JIA MAS, but other MAS cohorts, tend to have heterozygous single copy mutations, often amino acid changing, often rare, in these perforin pathway genes. And most recently, in collaboration with my boys in Philadelphia, Eddie B. and Scott, my mouse doctor colleagues, we showed some patient mutations in this gene we think might be novel gene in this pathway, DOC8. And the mice, where you deplete that, get a cytokine storm-like syndrome, not as severe, but similar in some ways to like the perforin knockout mice, for example. So I'm not going to talk about this because this is Mariana's work, but I'm looking forward to it. So on the left there are the HLH genes, and we've included DOC8 there as a possibility, and on the right is basically this pathway. So the yellow cell on the top is either a natural killer cell or a CD8 T cell, and one of its functions is to kill off infected antigen-presenting cells or target cells. And if you're defective in perforin or any of these genes we've talked about that are listed there in red, it's going to affect that. And so getting to Rashmi's question, I'm not going to talk about the work from our colleagues in Japan on IRF5 or Mike's work on IL-1RN, and Mike's going to give you a whole talk on HLA-DRB115, but I'd like to convince you that some of these HLH genes are probably relevant to kids who get macrophage activation syndrome, whether they have systemic JIA or not, but certainly relevant to this group, yes, for systemic JIA. But just because you find a mutation, even if it's an amino acid change, doesn't mean it's causative or contributory. It may be a risk allele of whether a child with systemic JIA is going to get MAS over and over again. And if it's a rarer mutation, it's more likely to be potentially pathologic, or if one of these algorithms like polyphen-2 suggests that it disrupts the protein function, that may be good, or if this particular mutation's been published in other kids with MAS of any form, really, for that matter. But what my lab does, and which is a lot of tedious, hard work, is try to demonstrate these individual mutations in vitro affect NK function. But that's really, you'll hear much more from Mariana and Mike about the rest of the genetics, but in terms of the HLH genetics, it's still more kind of a learning game. And I think that's it. And so I'll leave it to Mariana.
Hi, everyone. Oh, the slides are here too. Awesome. So I'm Mariana. Thank you so much for inviting me here today, and I have no disclosures. And one thing that was said over and over during this meeting is like, well, SJIA does not run in families. What can we learn from genetics? And we actually can learn a few things from genetics in general. So we can start by using genetics to see who is going to be at risk for developing a disease. We can use genetics to see if we can predict who is going to respond to treatment. And we can also use genetics to decide or identify people who might be at risk of having more severe complications or a different disease course. So starting by the risk of developing the disease, we have, oh my God, this looks, okay, well, I don't even know how that's gonna look. Okay, that's all right. So we normally think of it in 3 different buckets. So we can have variants that are pretty common in the population, and they increase your risk a little bit, like a nudge towards developing the disease. You can have some variants that are low frequency or somewhat rare, and they have a medium impact, so it's more like a push towards disease. And we have the other end of the spectrum, which is the really rare variants that have a big impact, like a tackle, like the monogenic forms of disease. So classically, we started studying adult SJIA by looking at the common variants, because we knew it didn't run in families, so this is the first place where we looked for. And these were the first studies where people were looking at genetics of SJIA. They were looking at candidate genes that they thought would make sense, like, oh, IL-1, IL-6. And at first, they did find some of the studies that found associations on those genes. And that actually drove very important studies and ideas that actually helped move the field forward. But those studies were small, and some of them were not replicated. So in some cohorts, they found association, and then in other cohorts, they couldn't find. So you see here, for IL-1, you have 2 positive followed by several negative ones. So then people were like, okay, so how can we tackle this? So there was this international consortium where they're getting people from many different places to do a genome-wide association study. And what did we learn from that? We actually learned quite a few interesting things. So first, we found that the strongest association with systemic GIA was with HLA-DRB111 and MHC class II. And that was one of the first things that actually implicated the adaptive immune system in SJIA, because up until that point, people were thinking like, maybe this is more like an autoinflammatory thing. The other thing was able to identify that the genetic structure of systemic GIA was different than other types of GIA. And finally, Dr. Grum did mention they went back to look at the candidate genes to see like, well, can we replicate some of those findings in this like multiple different populations? And they did indeed find that IL-1RN, that codes for IL-1RA, was significantly associated with SJIA. And in particular, it was a phenotype that was associated, well, a genotype that was associated with a lower expression of IL-1RA. And then there was this other abstract, still like pre-publication, where they found an association with CXCR4 with systemic GIA that was associated with neutrophil and monocyte migration as well. So now, looking about low-frequency variants with medium impact, now talking about the work that we did. So we had a large cohort of patients with systemic GIA with and without MAS, and we were looking for variants in HLH genes comparing to healthy controls. And we did find a significant association with HLH genes, especially SCXBP2 and UNC13D, with SJIA in general. Like, as expected, we did see that patients with MAS did have a higher frequency of HLH variants. But one thing that was very interesting is that even when we looked at the bulk of SJIA in general, we did see that those patients had an enrichment of variants in HLH genes compared to controls. And we also found that patients with SJIA often carry 2 or more variants in HLH genes more often than controls did. And interesting, in our cohort, we didn't replicate the A91V association. We actually had a lower frequency of patients with MAS having A91V than actually our healthy controls. So that was still like a little different than what had been seen before. Okay, so now getting to the rare variants. And I know that this is normally the thing that people think about genetics, but unfortunately it's the area that so far we have the least to talk about. The area we know the most, there's this family that they found in different families, this homozygous variant in LAC1. So they had these patients who showed up and they had fever, arthritis, the maculopapular rash that kind of really met criteria for SJIA. They did have a few things that were a little unusual, so they had a higher percentage of positive ANA and rheumatoid factor. And they did confirm that the segregation of the phenotype with the genotype associated with this homozygous variants in LAC1 gene. But later, the same variant was also found in patients who had inflammatory bowel disease with arthropathy, and some other variants in LAC1 were also found in patients who had other types of arthritis that were not necessarily systemic GIA. So it seems that LAC1 has this phenotype that is the severe arthropathy that may resemble SJIA, but I don't know if I would call it like fully a monogenic form of SJIA because it has a little different phenotype. There have been some other genes that have been proposed to be causative of SJIA with some case reports. There was a case report of a caspase-1 variant in a patient. That was paternally inherited. There was some functional data, but it was an N of 1. We have an abstract at DCR if you want to check in, that we found a patient with homozygous IRAC2 variants that we think are very likely to be driving disease in that patient. And one thing that Dr. Schuler mentioned, and I think a lot of people here have experienced, if you're having a familial case of SJIA, make sure you're not missing some other monogenic disease that can really look like it. So there have some case reports of MEFV, TRAPS, even H syndrome, and others that initially presented and met criteria for SJIA, but ended up being a monogenic disease, like another known monogenic disease. And now talking about treatment response, the same paper that identified the association of IL-1RN with the risk of developing SJIA also found another interesting thing. So if you had the series of SNPs that was associated with a low expression of IL-1RA, you had a higher risk of getting SJIA. But if you already had SJIA, if you had a group of SNPs that was associated with a high expression of, like high production of IL-1RA, you actually did not respond to anakinra. This study was replicated by an Italian cohort, but later there was a German group and a Dutch group from Dr. Vastert's here that they didn't find that association. One interesting thing is that going back to that figure where we were looking at the association of the IL-1RN with the risk of developing SJIA in each of the populations, you can see that the German group was the only one that didn't really have that association. So maybe there is something a little different about the genetic background of that population that might not have that association, so it still remains to be seen. And in terms of disease prognosis, so Dr. Cron already mentioned about the HLH variants with the risk of getting MAS, so I'm not going to get into that. And I know everyone is really excited to hear about Dr. Ombrello, so I'm not, talking about HLA-DR15, so I'll leave that to him. And that's it. Thank you so much.
Hi guys, thanks for giving me a chance to talk to you here today. I'm going to talk to you about HLA-DRB115. This is a picture of the Gaelic tradition boogeyman, and so with the relationship of HLA-DRB115 with bad things in systemic JIA, the question is this the boogeyman or is it actually a harbinger of potential opportunity? I have nothing to disclose except for the fact I'm going to talk very fast because this is a big topic and 7 minutes is probably not enough. So the first question is, what are human leukocyte antigen proteins? These HLA proteins are a family of proteins that are expressed on the surface of antigen-presenting cells to display fragments of proteins or peptide antigens in a standardized way, like a scaffold, that allows T-cell receptors to do immune surveillance of these cells, right? So the genes that encode these are located on chromosome 6 in a tight cluster of a lot of important immunologic genes. One group of these genes, see, I can't see that. So one group of these genes on the right of that picture, the class 1 region genes, these regulate the cytotoxic T cells and express antigens to them. And on the left side, you see the class 2 region. These are the genes that express peptide antigens to CD4 helper T cells. The interesting thing is in the middle between those two groups, there's a group of the class 3 HLA genes. This is a chunk of very important immunologic genes that are densely populated in that region, and all of these are in linkage disequilibrium, meaning that there's big co-inheritance of these gene segments with one another. You don't inherit, a small segment of 10 or 20 or 50 kilobases within the MHC region. You inherit chunks that are, 750 to 1.5 megabases in size. There are large ancestral haplotypes. So what is HLA-DRB115? So HLA-DRB, oh, the animation didn't work. HLA-DRB115 is one of these class 2 HLA alleles, which are the ones that present antigens to CD4 helper T cells. HLA-DRB115 is an allele group that refers to specific alleles, HLA-DRB11501, 02, 03, 04, and that group of those 1501 through 1504 make up a serologic group called DR15 that's an ancestral group. In fact, this DR15 is A, the most common ancestral haplotype, and B, the longest, one of the longest ancestral haplotypes in humans, extending in some cases up to 1.5 megabases. It's co-inherited with this DQ6, which is a group of the DQ class 2 HLA alleles, and in some cases, it actually goes all the way to HLA-B and HLA-C. And so in these extended versions, 90% of HLA-DR15 haplotypes actually goes all the way to the complement C4A and B genes, and 2/3 of them go all the way to HLA-B. So this is, DR15 is where we found the association, but the truth is this links to a large haplotype that could envelop a lot of other genes that might not be HLA-DRB115 as the effector of this risk. DR15 is immunologically relevant. It's the strongest risk factor for multiple sclerosis. It's a pathogenicity factor for Epstein-Barr virus, and actually it kind of triangulates Epstein-Barr virus and multiple sclerosis. It's also associated with a number of different autoimmune diseases, and it's been associated with drug reactions to augmentin in the past. So what's the relationship between HLA-DRB115 and systemic JIA? We started talking about this because of lung disease, but we've got to go back to the beginning now and say we're talking about first systemic JIA. And so to do this, we went back to the InCharge Consortium study, which was 9 independent populations, cases and controls from different countries that we then ancestrally matched genetically to make sure they were the same. Because when you do an association study, if you have cases and controls that aren't ancestrally and genetically the same, the difference that you identify is going to be the ancestral difference and not actually the disease difference, right? And so it becomes a mistake. You make an errant assumption. So in this particular case, we matched each of these genetically to make sure that we were making a proper comparison. And on the right two columns, you have either the allele frequency, so that means you take all of the chromosomes from all the people and you say how many of them have this allele. And you see there's not really a big difference between the cases and controls. And in fact, it flips between some of the populations. In two populations, it's more common in the cases, and in two populations, it's more common in the controls. And so in fact, this doesn't really look like it's associated with systemic JIA, at least from this perspective. We also have unpublished data, forthcoming immunochip study that Mariana mentioned. And in this study, we went back to the MHC again, because the immunochip has much more dense genotyping within the MHC locus. We have much better data in that region. And it's very clear that HLA-DRB115 in this study wasn't really associated with systemic JIA as the phenotype. So stringently controlling these. The reason this is really important is because HLA-DRB115 and all HLA alleles are stratified across populations, and even in these countries in Europe, the population frequency difference. So you can't take some people from Spain and some people from Italy and compare them to one another, right? In Myanmar, the frequency is as high as 84% of people carrying HLA-DRB115 alleles, and in South America, there's populations where nobody has it. So there's a wide range. And in the United States, look at the difference between the Asians and the African Americans and the Europeans and the Hispanics. It's really difficult. You can't really just aggregate people of different ancestries. And the reason I mention this is because we're trying to figure out why there are two studies that have shown that there's a much higher frequency of HLA-DRB115 in systemic JIA than has ever been seen before. There's a few reasons this could be, and we'll talk about this when we have the panel discussion, I think. So, kind of the big question, what's the relationship between HLA-DRB115 alleles and Still's lung disease? So, in the study that we published, we found that in a cohort of people who had Still's disease, lung disease, which was mostly systemic JIA and a few adult Still's patients, 82% of them had HLA-DRB115 alleles. When we compared that with the in-charge cohort, we found that the odds ratio for lung disease was 14.1. So it was significantly higher in the people with lung disease than in just the standard systemic JIA patients. Then we took a group of systemic JIA patients who had received the treatments that we've been talking about and didn't manifest lung disease or any features of hypersensitivity. So we called those drug tolerant because they had received the drugs, they had responded, and they hadn't had any negative features. And in comparison of that group, we saw that the odds ratio was about 58. And so that suggests even more. This is a pharmacogenomic study now at this point, right? And so we've gone to people who received the drug and didn't have what we thought was a reaction to the drug and compared it to people that we thought were having some sort of a reaction to the drug. So since we published this study, multiple different groups have published their findings from their cohorts as well. So we have single center studies from Minnesota and from Boston and from Rome and from Utrecht and from Cincinnati. And while I don't have time to talk about all of these things, the critical thing that I did was aggregated the lung disease data from all of them. And so within the lung disease patients, 84% of the lung disease patients across these studies have HLA-DRB115. Now, when Grant and Vivian's papers were first written, there was conversation about features of hypersensitivity that didn't make sense. And the one that really hammered home was this anaphylactoid reaction to tocilizumab. I mean, that's a hard piece of data. And we didn't know why it's happening, and we don't know why it's happening, but you can't deny the fact it was happening. So we wondered, after we saw this association with HLA-DRB115 and lung disease, whether there might be some people that were having these features that were atypical that weren't having lung disease. So the people in the study went back and looked in their cohorts and indeed found groups of people who were having hypersensitivity reactions, features of hypersensitivity with these cutaneous eruptions, either immediate type hypersensitivity like anaphylactoid reactions or delayed hypersensitivity like 3 to 7 day eruptions of diffuse urticarial lesions or even erythema multiforme type lesions. So first, the people who had the drug reactions without lung disease had 82% of them had HLA-DRB115 alleles. This is about the same as what we saw in the lung disease patients. When we looked at the people with no lung disease but who had had a reaction to the drugs and we compared the frequency to in charge, we saw a pretty similar odds ratio to when we used lung disease as the phenotype. It was 11. And then when we used the group of people exposed but without reaction or lung disease, the odds ratio was again similar to what we saw with the drug tolerant controls. It was about 55. So this is a unique combination of immediate and delayed type hypersensitivity that, you know, it doesn't comply with any sort of Coombs type hypersensitivity reactions that we've observed in the past. So when we went back to the cohort studies again and we looked at what they had found, now I took a liberty here of taking the drug reaction, the DRESS, the fixed rash, the drug reaction, the drug reaction, and the drug reaction and consolidating them down. And then took that group of people and said, what is the HLA-DRB115 carriage among them? And again, we have 85% of them who have the HLA-DRB115 allele. So on one hand, we don't have data, right? We have hypotheses and we have observations and we don't have data. But we can take the data we have and try to dig deeper into it and see if we can glean additional information that at least points us in the right direction. So with these observations in hand, we now start to wonder, what is it that's happening here, right? We have these patients with systemic JIA that are getting medicines that other people with other diseases are also getting. And in the context of getting those medicines, they're having features that look like hypersensitivity reactions. Their systemic JIA is getting worse and they're getting lung disease, right? And so one possibility is that they're having a drug reaction. It's linked to HLA. HLA-associated drug reactions is a pretty clear thing in the literature. And so the first thing on the list is that it could be related to presentation of pieces of those drugs on the MHC molecules to T cells and causing an immune reaction. Problem is, there's 3 different drugs and there's no molecule that's in common across them. There's a thing called a hapten reaction. A hapten reaction is where something in a drug, the medicine or something else, changes the shape of things that our body normally presents. And when it does that, something that my immune system might normally tolerate might be shaped differently. So suddenly my immune system could theoretically recognize that. That's a potential possibility that could be happening here as well. You could also be having a reaction to something else in the drug that's not necessarily the active ingredient. But as everyone knows, we could also have some sort of immunologic voodoo happening with this accentuated IL-1 and IL-6 and IL-18 signaling, and we start to tinker it by severely decreasing the IL-1 or the IL-6 and then leading to a Th2 shift. That's another very plausible hypothesis. In addition to that, there could be an existing infection or an emerging environmental exposure, a toxin, for example. And of course, there could be something beyond the bounds of my imagination. The thing that I'm thinking a lot about is whether there's something else on the HLA-DR15 haplotype. And I think that's something that's worth investigating. So if we flip this around and think about the types of mechanisms in a different way, you could have antigen-dependent type mechanism, and that could involve pieces of drug, pieces of virus, pieces of altered self-peptide, or you could have an antigen-independent phenomenon. And this could involve something like, cytokine plasticity and Th2 shifting in response to our treatments. Could also involve something like a non-classical HLA gene someplace else on that haplotype. So I've talked a lot, but I don't feel like I've told you very much because we don't know what's happening. But the key is that it's become controversial in the field, but it doesn't have to be controversial because there's a list of possibilities and we just need to look at the list and think about which one it is and generate data to move through and cross the things off the list until we find the hypothesis that's correct. I sit up here on the 13th floor of Building 10 at the NIH and I think about Still's disease all day. If you guys ever want to come visit me, give me a call. Thank you.
So, Karina Dagor is asking, what is the genetic test can be performed to assist in diagnosis of Still's disease, if there is one? And then Lauren Henderson is making a comment. So there are patients who present eosinophilia and pruritic at disease onset without exposure to biologics. Same for LD.
I'm going to take a point of privilege because I have a captive audience. Rafaela and Betty Diamond wrote me into editing a particular issue of Frontiers in Immunology, have a mid-impact journal, impact's around like 5.7, to put out an issue related to MAS. I broadened it to cytokine storm syndrome, so if it's HLH, that could be included. So if anyone in here and/or knows someone who wants to put out something this coming spring on that topic, please contact me. I'm easy to find, my email or phone number or whatever. Sorry.
So in terms of the eosinophil count, this is something that I didn't really touch in my talk. And the reason I didn't touch it is because I think as a community, before we start to address it, we need to agree on the thresholds that we're going to use to define the abnormality, and we need to define the time points or the ways that we're going to look at it and track it over time. And then we need to systematically evaluate that in more than one cohort. And in doing this, I think that it's going to help us to better understand what's happening with this development of eosinophilia. Because while when we go to the margins and use a threshold of something like 500, we might find differences between the groups. When we look at the actual reactions that are classifying as reactions, those patients don't just have an eosinophil count of 500. And the other thing that I want to point out is that a composite measure is always better than a single point measure. I think I heard people say that 7 times yesterday. And in this particular instance, when we look at the features of hypersensitivity, it's a composite measurement. It's not an individual thing. We don't have a person with isolated eosinophilia that actually classifies. They have itchy rashes, they have eosinophilia, they may have elevated liver function tests. But in the end, these are my observations, and I think we all need to publish our observations because what I see might not be what everyone else sees, and what I see might be completely wrong. So, Lauren, what you say is very valid.
I would just add, you put patients at new onset, like for profound eosinophil spray. They have been good. Not all patients are good. The number that you picked up.
I mean, I think that's true. My most recent, as a teenage boy with systemic JA, who had not seen any drugs, including non-steroidals, had 30% eosinophilia and a normal white count. So lots of eosinophils, and he had not seen any of our drugs. So at least for that child, and for the ones that Lauren's mentioning and others, eosinophilia may just be part of the disease in a cohort of our kids. That's not to say that drugs can't also cause eosinophilia, but I think it's important to recognize that every time we see eosinophilia, it's going to be hard to ferret out why that is.
Dr. Cron, I think Dr. Cron was going to ask us, did you see this before? Like, I know I'm pretty young, so I've been treating pediatric rheumatology patients since biologics have been around, and not only biologics, but this phenotype of lung disease is relatively recent. From people who were around before the phenotype emerged, did you see people showing up with disease onset with eosinophil counts of 12,000 at onset?
Did you evaluate them for parasites?
Yes, actually, we saw those patients before, and actually Earl Brewer published a large paper on that in 1976, describing specifically eosinophilic expansion at disease onset. But I wanted to ask you another question. I was actually finishing a chapter on systemic juvenile idiopathic arthritis and was sort of working on the section describing the classic Still's disease rash. And then I started thinking, when was the last time I saw it? Most of my new patients in our clinic with new onset systemic juvenile idiopathic arthritis present with this bizarre fixed pruritic rash. And we actually biopsied this rash a few times that looked to me a little bit like neutrophilic dermatosis. So the point is that in Cincinnati, certainly we see a shift in the phenotype of the disease in general, but the rash in particular. We're seeing more and more patients with this pruritic rash. Pierre Cartier in Europe published a very interesting paper describing the presence of this rash in a large proportion of their population, and the presence of that rash at disease onset actually is a risk factor for recurrent MAS, refractory disease, lung disease, and so on. So I just wanted to ask other people in the room, are you seeing the same shift in the phenotype of the rash?
I don't know if there's a shift, but I'm old enough that I was one of David Sherry's early trainees. And actually, the very first kid I ever saw in the hospital as a fellow, as a consult, was a young boy with systemic JA. But he taught me back then, because it wasn't in the textbooks and wasn't in the literature, that a lot of these kids have these fixed type rashes in what we call systemic JA, which I still think is a relative gamish. They're not all the same, but they do share common features, and they often respond to common therapies, so we treat them the same, call them the same. They're clearly not all the same, but I think this has been around.
I just want to add that too, since I'm one of the older generation now. Yeah, we definitely eosinophilia, itching, and different type of rashes. And I agree with Alexi, there might be some shift in the rash. Like this salmon color thing, rarely now we see almost.
Michael, can I catch your attention? This side, yeah. Yeah, interesting work. So basically, I'm wondering that you mentioned the significant association of HLA-DRB115 with lung disease and the drug reaction. So do you want to make a case that all systemics should be screened for this particular HLA and the clinical implications of this?
Well, so my feeling is that everyone that I see is going to get screened for this. And if I were practicing in an insurance-based situation, I would be trying to bill for this in my patients as well. I make the recommendation that we screen for this. The question that comes back to me is, what do we do after we find a patient who's got the presence of
And
that
before
allele,
that, because
right?
in one of your slides you mentioned that the prevalence is quite variable. For example, Asians, you showed it to the tune of 40%.
80% is the biggest Myanmar
Yeah.
population, but yes, 40% are pretty common in the Asian populations.
So how do you go from there?
So as this has evolved from its original recognition and the alarm of the situation to observing patients over time, and now at this point we've observed a lot of patients who've come through our clinic, which gives me the opportunity to see how all of you manage the patients and to see how they respond to the things that you do. And in the end, my position now is that if the patient doesn't have any of the risk factors that are concerning, I'm okay to initiate therapy with one of the standard therapies. But if there are red flags that start to manifest, or if the patient isn't brought into remission with one of those medicines, my feeling is that I would rather move to other medicines and try other things for which I'm not concerned about this risk. This is an individual approach at this point, and so I'm just sharing with you my approach, and I'm sure that there's many people who have a very different approach than me.
But I'm not sure the genetic effects side, right? Because if you had a patient who was getting clubbing or whatever, whether they're DRB positive or not, you're going to make a change or not, but that's going to probably affect your decision more than an allele that's pretty frequent.
I would certainly hope that I would identify something other than clubbing as I'm looking at the LFTs, the LDH, the eosinophil
Yeah, I'm
count,
not
looking at
saying
this- No, I'm saying
you're going to find it late. I'm just saying
anything.
there's going to be clinical stuff that's probably going to drive you more than the actual genetics, I would hope.
So there's 17% of people who don't have HLA-DRB115 among the patients who have either these features of hypersensitivity or the lung disease. So in the end, the absence of the allele, you're right, isn't going to stop you from changing course on therapy if you have a patient who starts to manifest these sorts of features. If you're a person who does this, if you're a person who adds another drug or a second drug or a third drug, then that's the thing you're going to do. But for me, if they start to get features, I'm probably going to stop the drug and change to something else.
I have a comment, and then I'm handing it to Boss. So one thing is just the moving target of what we even mean by systemic JI stills. Patients with monogenic diagnoses who may have presented 10, 15 years ago might have been lumped in because we didn't know about those genetic diagnoses, whereas patients who didn't have arthritis probably were excluded, which probably was enriched for a bunch of these eosinophilic fixed rash patients, but we're now including them based on the work of people like.
You're right. You're right.
Really interesting work and also an important discussion, actually. So the point here is, how much time does it take for you to do the genetic testing? Because we know that starting early therapy really affects your response to therapy as well. So that's for one. And the second thing is that indeed, and I fully agree with the other people here, that the prevalence of this haplotype is so frequent that by delaying this and putting this as the main risk factor, you're putting at least a significant number of patients also at risk for disease aggravation as well.
So I'm not sure if I'm putting them at risk. I think the information that we can gain from HLA typing is important. For us, we get a turnaround of HLA types in about 2 days from when we send them to our clinical HLA lab. If we're going to do the sequencing and extract it, then that's going to take 2 months. So we send them all clinically now at this point. I'm not recommending that we delay therapy necessarily. I think early therapy is important. when I see them for the first visit, that would be one of the labs that I checked.
But this is also important, Mike. So in a new patient that you see in your clinic, you're doing HLA typing. We do as well, but we do it
I am.
hindwards.
Say what?
We do it behind or afterwards. But then you, how do you treat an HLA-DRB? Because it is prevalent. So you, at least 1 in 4 of your patients will come back with HLA-DRB1 carrier.
Ours are close to a half because we have an enrichment of patients who have refractory
you stratify
courses.
then the start of treatment based on your, on this haplotype? Go ahead.
So, Dr. Vasta, like, we have a little bit of a benefit of being a referral of a referral of a referral center, that normally the patients who come to us, they've had disease for a while. And what our rule of thumb is, if things are working, keep working. We're not changing. If these are not working, then we think about what's going on. And I think that the HLA-DR15 in that context is, we're not going to say don't give IL-1 inhibitors, because we have seen patients who had the haplotype and did fine, and they're doing great, and we're not going to touch their therapy. But if we know that they have the haplotype, we're going to have a higher suspicion, and we're going to look at them closer, because we also know that those patients are at higher risk of having refractory disease, to not respond to medication, to have reactions. So we would just have a higher index of suspicion. And actually what we end up doing is very similar to what the Boston group recommended, of like, you look at the risk factors. So, and then you kind of go from thinking from there. And even like before I came to the NIH, I was thinking about one case where that changed my management. So it was a boy who was sick, like he had disease onset at 6 months old. His IL-18 level was greater than 100,000, like I think to 180,000. When we gave him tocilizumab, he developed this persistent itchy rashes that were fixed, that were like very, very, very unusual. And at that time, I didn't even think about it. And he had a history of congenital diaphragmatic hernia. He had persistent elevated eosinophil counts. And when we were trying to give him anakinra, we were going on high doses, and he was not responding great. So he had anakinra tacrolimus. He was like needing a lot of steroids. So when we saw that that patient had an HLA-DR15, we're like, he's adding up a lot of risk factors here. So that was a patient that at that time we decided to switch to a JAK inhibitor. So I know that he already had a lot of risk factors going, but I think that when you add an HLA type to that context, I think that adds your index of like what you're going to do. Of course.
From a parent perspective, first, as the parent of a child with systemic GIA, with the positive HLA type, the first thing that happened to him when they discovered the lung disease was they took him off all the medicines, anakinra, methotrexate, everything that he was on, because they were afraid. And within 10 days, he was in MAS in the hospital, and they could not control the MAS. They just could not control the MAS. Till I remember one morning, our doctor said, I think we have to bring back the anakinra. And they did. And, I think when you have these patients, these very complex patients, the expert physicians, even if there is a risk to the biologics, I think your research is about the HLA type is brilliant, and it has really impacted the field. And maybe there is a risk to the biologics. But even if there is, the treatment of these patients needs to be done by an expert physician who can use every tool and do this delicate dance to keep the patient out of being in the hospital all the time, and giving up the primary tool that we have in this disease, which is the IL-1 and the IL-6 inhibitors. Even at this time, they are the best tools we have, is really depriving that patient of opportunities. And I really think this is strongly, we've seen this play out in many, many parents. And yes, JAKs are there, and we are seeing increasing usage, and it's a good thing to have options. Some patients do really well on them. Some are able to stop the biologics, some are not. But the disease control in JAKs, and I wish we'd gotten to that more, but we haven't, is actually different than the disease control that can be achieved by biologics when biologics work really well. When biologics work really well, you pretty much go back to a normal, like normal life. With JAKs, you really do have more immunosuppression. We are seeing more infections. I think in the group, we are talking a lot about parents who are saying that they are seeing growth issues, that the kids are not growing the way. I think we need to understand the JAKs before saying, stop the biologics and move on to the JAKs. And so I really, I worry about a strong message going out in the field that, don't stop the biologics and what it takes to actually manage a child who's this incredibly complex disease that we talk about.
I have one question here.
I just wanted to make one comment, Rashmi. So when biologics are working, we're not stopping biologics. So that's our rule of thumb in terms of if things are we don't change the thing that's weaning. So if we see that the person is responding to biologics, we don't touch. And actually now at the NIH we don't do anything because people come to us when they're already having the therapy. Sometimes we can make recommendations which they might follow or not. But we have a colleague that actually works with MAS. So he gets all the referrals for the crazy MAS that are happening in the country. And he started reaching out to us because he's seeing a few patients. I'm not saying that's the rule by any means, and I'm not saying that this is the right thing. But he's seen patients that when we plot the course of how they're going, you see that as they're going up on the anakinra, the patient was getting worse. So I'm not saying that this is the truth for everyone. I'm not saying that this is the fact for everyone. But then we see that the patient just have liver enzymes that shot up to over 1,000, which is something that has been known for a while, that some patients do have liver injury from anakinra. So we're just saying that there are patients out there who it's for them, it doesn't work as well. So that just to keep an open mind that that might be there are patients where in their case the biologics might not be doing the best thing. There are patients we definitely keep them on even regardless of the HLA type.
I worry we fail those people. If they're not doing well and we don't change, I feel like we're failing those people because our job is to be the expert who moves to the next. We fail all the time.
We have seen cases in the patient group. I'm sorry to be so blunt, but we have seen cases when, and I apologize if I'm being blunt, but this is over many years. We have seen when patients were taken off biologics and it did not go well. And at least biologics were keeping them alive. I think we need, which is why the foundation's work is really focused on finding other therapies. Because I get your research. I think, as I said, I think it's brilliant, but we can't just take away the tools. I think that's
There was a question on Zoom at the beginning that no one answered, so I thought I'd maybe just... the question was, what genetic test, this is from Zoom, not from me, what genetic test do you do to diagnose Still's disease? And to me, there is a role for doing genetic tests, but it's not to diagnose Still's disease, it's to make sure you haven't got the diagnosis of Still's disease wrong. So there's other autoinflammatory diseases. I find the arguments about DRB1*15 interesting. My overall take on this is that it's possibly a relatively mild risk factor for disease severity, but probably has very little to do with the propensity to drug reactions. And I find the arguments about whether or not you base your clinical decision-making knowing about the DR1*15 perhaps less persuasive, because what you've said is true, but you're going to make the clinical decisions irrespective of whether you know the HLA status.
We are, but the concern is that some people aren't. And so that becomes an indicator that tells people who might not be aware or might not be in tune, you got to pay attention here, right? I'm not concerned that you or Boss you or Alexi, right? But we're talking about communicating with the community as well.
That's exactly the point, Mike. Sorry. Can I just... That's exactly the point. So I think listening to your talk, we have more in common. I really appreciate your work, and I do understand the close association of HLA-DRB1*15 and at least lung disease, because these data are compelling. But the point here is that in the publications, this nuance is gone. So you are telling our patients that we see are really the refractory patients. And of course, I understand that you type them and you wait that in many other risk factors in a decision on how to proceed on treatment that is that has proved to be refractory or difficult to treat already. But the SJIA paper, in which you are second author, is is actually concluding with taking this at first-line treatment into account. And this is really a difficult message to understand for many of us, and many of the doctors in the field, I would say.
And this is why I'm trying to fix this through my comments in public, right? Because there was some disagreement about what the final conclusion and what our response should be to identifying patients having this, right? I wasn't necessarily on the extreme within our group, but we compromised as to what we said. The reason we used the instrument was because we wanted something that was standardized to talk about a drug reaction, because when somebody just says there's a drug reaction and this is this or this is a rash or that, it's not really objective. But at the same time, if you can use a composite measure like an instrument, then suddenly you can show that there's something else that's happening. But I think that there's also other genetic factors that are at play, and we can talk about this at another time. HLA-DRB115 isn't the only thing. We talked last year about the interferon signature and genetic factors that are underlying that. I hope that we'll have something that we'll submit here within the next month or two, and then you guys will be able to see that specifically. It's not, HLA-DRB115 isn't the only thing by any means. I think that there are systemic JIA specific factors, and we need to figure out how the medications, whether it's the biologic effect of the medications or something else that's kind of contributing to this new emerging phenotype.
May I bring it back to the point about tools and new therapies? So I represent a company that is interested in potentially developing a compound, completely new mechanism of action in Still disease and MAS. So would you consider this patient population with this genetic marker, and I come from oncology, by the way, as a potential patient population where new drug development would be relevant, and then potentially a companion diagnostic associated label?
What was the last thing you said? I'm sorry.
So in oncology, we develop, of course, drug with companion diagnostic, right? You have the mutation, you get this drug, including in first line, by the way.
So I'm exceedingly interested in genotype-driven therapy for the patients and using genotypes to identify therapeutic opportunities. I think HLA-DRB115, so we see these associations that I mentioned, but in the end, you could take a step back and you could make a different conclusion like what I think Boss has made and others, which is to say, really this is tagging a refractory to treatment group of patients, right? And so yes, this is the group of patients in whom the need is the greatest among Still's disease to identify a new therapy. I think that this marker could theoretically define a severe group of patients.
So, sorry if I take a little bit. You were mentioning NK cell at the beginning. Yesterday I was having a brief conversation about NK cells. So again, I come from oncology. I work in the CD38 field. Of course, daratumumab completely obliterate NK cells. Literally, we just developed some initial data a few days ago, which seems to indicate that our molecule preferentially deplete NK cells, at least in RA patients. We haven't got any cells from Still disease patients. If somebody can give me some, or I give them the drug. But anyway, would all of what was discussed just now, is it related to the NK cells and the innate system, or it seems to be, anyway, please help me out.
That's a good question. That's a good question.
I mean, I don't know. The SJIA is thought to be autoinflammatory / autoimmune. Early on, autoinflammatory, and then over time, as the arthritis kicks in a subset of patients, it's more autoimmune. But in the NK cells, at least in terms of the macrophage activation syndrome, you want them functional. So you would not want them gone.
I would imagine, sorry, I have a question now. I would imagine that several siblings of patients with SJIA have the same genetic background, the same IL-1R and polymorphisms, but they don't develop SJIA. Why is that? And then as a follow-up question, what are your thoughts about, so I'm a BMT physician, and in general, we prefer to use a matched sibling donor if possible. And so if there's a donor within the family who's a match, and we've done all the genetic testing, there's no genetic variant or whole exome, or if our whole exome sequence is negative, is it okay to use a matched sibling donor for transplant?
So thank you for the question, and also I apologize for the question on the chat from earlier about what is the genetic test to diagnose this disease. I think it's in a similar way that the associations that we've found so far, they are genetic risk factors, like a nudge. They're definitely not fully explanatory of the phenotype. So there are other complex environmental or genetic things that play a role in the risk of developing. So we still don't see a strong enough risk factor genetically that would start seeing it running in families, right? But the question of the matched sibling donor, the issue is not because of HLA-DR15 or because of IL-1RN or HLA-DR, but we believe that there might be some true monogenic cases that we still don't know the gene, or oligogenic cases. So, for example, we have we do have this patient with IRAC2 that has homozygous variants that we think might be pathogenic. So, if the age of onset is super young, there's always the risk of what don't we know that could be going on in here. So I think that is it's a risk.
Just to follow on what you said, Dr. Marquez, so the patient with the homozygous IRAC2 mutations, right, doesn't have siblings with the variants, but both parents contributed one of those. Both parents have an intermediate metaphenotype. They don't have Still's disease, but they have an intermediate immunologic phenotype that is stronger in the person. We have other instances with siblings who share metaphenotypes without Still's disease.
I imagine we should be okay if we're able to do, like, say, whole genome sequencing and, make sure we're not missing a monogenic variant.
So, I mean, I'm like, first, it makes sense. I think that just a lot of those genes are unknown because, we have been looking for those genes for quite a while, and we haven't found two people, we don't see many of the repeat genes, so which makes it a little harder to study. We see some genes clustering in certain pathways. So the thing is, you're going to get a list of genes, and there's going to be a lot of unknowns. And, unless that's the issue, it's just...
I guess as a counter argument to that, I would say that if it's truly monogenic, then we should have seen more siblings develop SJIA as well, right? I mean, we see it in other inborn areas of immunity that, multiple family members do get affected.
No, that is an excellent, the relatively common rare diseases that we're already thinking that, that's probably not possible, or not likely. So it's more likely to be either a de novo variant, if there is a monogenic form, like a de novo variant, or ultra rare, interaction, or it could be oligogenic of just the really bad combination of things. But you're right, the risk is relatively low, especially compared to other known diseases. I think that my main caveat, I'm not saying that you shouldn't do this because I know that from the BMT perspective, it's the dream. It's just, if the disease had onset, it's such a young age, it's inversely proportional, the risk of what are the chances that this was just driven by an environmental factor, right? It's just, I would just be cautious for the very, very young patients with a really severe phenotype. But I think that that's true for most things, I would guess, that are transplanted for, right?
I think there's one quick last comment from Bruno Fortville, and then we should
I'm coming back to the drug reaction, and when you explain that, I have the feeling that drug reaction for you is you have to stop the treatment, which is a problem because 25 years ago we had the same discussion with NSAIDs, with antibiotics when the people started their disease. So I think presently with a patient for which we have this question of skin reaction to anakinra or tocilizumab. For tocilizumab, for example, it's very frequent IV, but when we switch the patient for sub-Q, we don't have that in adults. So it means that it's not really an hypersensitivity to the drug. So at the end, I think it's better to say, maybe the drug has something to do with the reaction, but it's better to say not all reactions should lead to drug interruption. And you just have to add something, but not to stop the ongoing treatment, which could be very important.
Well, on that note, I think...
I was just going to say, I think there's substantial confusion out there because when we were diagnosed, anakinra was conditional upon DRB15 negative That was how it was presented to us. I'm not saying that that they would refuse it if we came back positive. I'm just saying that there's confusion.
Yeah.
It's substantial. So.
Well, I think ultimately the parent should have a say, and because we're choosing between a rock and a hard place.
Yes.
And so I think the parent should have a say if they choose to take this risk
Yeah.
for their child. Yeah.
I do agree. Sometimes the message is not as nuanced when it's being practiced out in the field. Okay, with that, thank you so much. We did address the elephant in the room.